not working now. Come on. There we go. So that's why this should look a little familiar. There are lots of gram-positive bacteria that cause disease. Again, there are lots of gram-positive bacteria that don't cause disease, but we don't really care about them in this class. And because there are so many gram-positive bacteria that cause disease, we divide them up based on their shape first. And then if they're gram-positive and they're cocci-shaped, then we start doing those tests, those same tests that we do in lab. Again, even if you have a catalase negative, that's still two really big groups of bacteria. Streptococcus, there's lots of species in strep, which we're actually going to kind of skip to. And there's lots of bacteria in the Enterococcus that cause disease. And later on in lab, we will do another test that divides those two out as well. There's also a whole bunch of gram-positive bacilli or gram-positive rods. But intro stuff. Intro, we talk about a little bit about staph. Most of them are normal flora. The only one that really causes disease is the Staph aureus, which is what we're testing you guys for. And that's because it has lots and lots of different virulence factors that allow it to cause disease. And we were testing specifically for the fact that it makes that coagulase enzyme, so it's easy to test for. Otherwise, it has lots of things that allow it to cause disease and evade getting detected by our immune system. I don't know why I had it swipe. Lots of different enzymes that it has and can make. It can also, it's naturally resistant to a whole group of antibiotics. So it could care less about any of the bacteria that target the cell wall. So some bacteria are just naturally resistant. It didn't acquire this resistance. It's just naturally resistant to any of those antibiotics that target the cell wall. And that's because it makes, I'm gonna go back, an enzyme called beta-lactamase. And so any of the bacteria that target the cell wall, that peptidoglycan, they're called beta-lactam drugs. Well, this makes an enzyme that just breaks down that drug. So that drug, like penicillin, is going to do absolutely nothing for these groups of bacteria. Luckily, there's lots of other antibiotics we have. I don't know why I had it swipe. It also has lots and lots of toxins, which is generally what's going to cause all the horrible symptoms and side effects from Staph aureus. And it can cause not just skin infections, cause food poisoning, as well as lots of horrible skin infections. If it can also get inside the tissues, that's what happened to Addie, and it can cause lots of other things if this bacteria gets into the body. Again, I'm skipping through, this was all in the intro. Easy ways to detect it. You can just do a gram stain, you do the catalase test, you can do the coagulase test, and it will tell you kind of, which staph you have. But again, if it was catalase negative, still gram-positive cocci, it narrows it down to Streptococcus or Enterococcus, which doesn't really narrow it down all that much. And there are lots of Streptococcus bacteria. Still in the intro, one of the easy ways to help start narrowing down which Streptococcus bacteria it is by looking at the different hemolysis reactions on a blood agar plate. Because there are strep that are beta hemolytic, there are some that are alpha hemolytic, and there are a couple that really don't cause any human disease that we don't care about that are also gamma hemolytic. But because there are so many Streptococcus species of bacteria, we group them based on little antigens that are on their surface. So it's not something we can visually see underneath a microscope, but it's how we group them just based on what antigens they have. And we say they either have, they're called Mansfield grouping, we either say they have group A antigens, B antigens, C antigens, D antigens, or GF antigens. Now, the ones that seem to cause the most human disease are at A and B. And so the very first one that goes through in the intro is the strep pyogenes, the bacteria that causes strep throat. And so when we were testing you guys for strep throat, we even put your bacteria on what was known as the SSA plate, which was a selective strep A plate. We are looking for any bacteria that would fit into this group A. And strep pyogenes is not the only one, because I think everyone had bacteria growing on their strep A plate, but it's a big clue that it might be pyogenes, especially if it had that beta hemolysis. That strep bacteria also has lots and lots of virulence factors, lots of enzymes, lots of toxins, lots of things that allow it to get in and cause disease. And causing strep throat is not the only thing strep pyogenes can cause. Can cause scarlet fever, big pus-filled lesions. I put a big old line on here is because there are different strains of strep pyogenes, and some of them can cause toxic shock syndrome and even the flesh-eating bacteria. So strep pyogenes doesn't just cause throat infections or strep throat. Other things it can cause. I think this is the last slide of the intro, is diagnosing strep pyogenes, which starts us off into our group B of Streptococcus bacteria. So it's still a Streptococcus. Again, it's just not a group A Streptococcus bacteria. It has B antigens on it. When we gram stain it, it's still gram positive. Everything in this entire lecture today is gram positive. It likes to hang out usually in chains. It's different from the group A Streptococcus because it has group B antigens on its surface. So when we do different types of testing, we can test specifically for B antigens. However, on a plate, you can't see any of that. It does beta hemolysis, just like pyogenes was, which is a group A. However, the beta hemolysis is usually a little bit smaller. Someone that has that strep throat infection, that eating away of the red blood cells is going to be very evident, huge clearing around the colonies. The strep B, the Streptococcus agalacti, usually has a smaller zone of beta hemolysis. It's also resistant to an antibiotic called bacitracin, which some of the other strep bacteria, which is coming up, are sensitive to. Now, Streptococcus agalacti, two E's at the end, and they're agalacti. Why we care about this particular bacteria is because, of course, it can cause disease, but the predominant group of individuals that picks up this, that it causes disease in, are newborns. And so it's usually considered a neonatal disease. That doesn't mean other individuals, older immunocompromised patients, are also susceptible, but the highest group of individuals that generally suffer from this particular bacteria are newborns. And it's because mom was a carrier of it. It didn't cause any issues for mom, but in a newborn that has very little immune system to do anything, it can cause bacteremia, it can get into their bloodstream, it can cause meningitis, pneumonia, it can lead to blindness, it can lead to developmental issues. It can cause death, but that's still also pretty rare in today's age. But it is predominant. I want to say it's not super, super common. Before we started doing testing and treating, it was about every 3 in every 1,000 births. Which you're like, well, that's not many. What do you think about? Do you really want to be that, 3 in 1,000? It's still like 1 in every 300 births. And you think about how many births take place just in La Crosse alone every month. Now, we can test for the bacteria very easily doing an ELISA. It's a very treatable bacteria. It's not developing resistance that we know of very much right now. So it's easily treatable with penicillin, ampicillin. But the biggest prevention is that mothers, before they give childbirth, they will test them to see if they're a carrier for this particular bacteria, as well as other bacteria, and they will give them antibiotics. So again, it's a bacteria that doesn't cause mom any issues whatsoever, but they will give her antibiotics to get rid of the bacteria, so the bacteria is not present before childbirth, because this is a bacteria that does like to hang out in the female reproductive tract. So during a vaginal delivery, that's when the baby generally picks up the bacteria. Easily preventable. And there's a whole group of Streptococcus bacteria. So the ones that mostly cause disease are in group A and group B. But then there's a whole nother group of Streptococcus bacteria that don't have A antigens, they don't have B antigens, they don't even have C or D or any other letter antigens. So we kind of call them they're like the misfit strep bacteria, but they're officially called the viridins group. So they don't have A antigens or B antigens or any other antigens that we normally classify our strep bacteria. So they just get their own unique group of Streptococcus bacteria. Anything in this viridins group of bacteria, they're all alpha hemolytic. So on a blood agar plate, they're going to have that nice greenish color to it. Most of them are normal oral flora. So you guys swabbed your throats like a week ago, and we looked at your blood agar plates, and I can just about guarantee All of you wrote down alpha hemolysis. I think you all did. On your lab report ones is, yes, I had some alpha hemolysis growing on my plate. And it's because it was going to be some of this normal flora that's found in this viridans group of Streptococcus bacteria. They generally don't cause disease, normal. However, they can be opportunistic pathogens, which means they could cause disease. And this is usually when they get somewhere where they're not supposed to, and you throw in some immunocompromised state as well. The biggest time that these cause disease, since they are already just part of your normal throat flora, is if you floss and you haven't flossed in a while and it bleeds, open wound sores for bacteria to get in. Again, if you have a healthy immune system, you can generally take care of it right away, but if you don't, you just gave those bacteria an entry into your bloodstream. And if you're immunocompromised, if it gets into your blood, it can cause meningitis, it can cause endocarditis. It could be deadly. Again, healthy immune systems, not really a big worry. Number one reason why people that have weak immune systems, elderly patients, if they are going to go to the dentist or have any kind of dental work done, they're usually given an antibiotic ahead of time so that any of the bacteria that do get into their bloodstream, they're already on an antibiotic to fight it off because their immune system can't. These are also some of the bacteria in here that hang out in your mouth, on your teeth. You probably saw some of these when you scraped your teeth because they are living there and eating all the candy that you just ate. They are breaking down those sugars, producing acid, and these are some of the bacteria that are the top causes of your cavities as well. As I feed you guys candy today. Another bacteria that's still in this viridin group that also causes disease is Streptococcus pneumoniae. And yes, its name is that it causes pneumonia, but it's not the only thing that it can cause. When we gram stain it, it's still gram-positive, it's still cocci, but it usually likes to hang out in like short little chains, sometimes even pairs. Alpha hemolytic. And so since it doesn't have those A or B antigens, it's still in this viridin group. And for a lot of individuals, this is still a normal flora bacteria. Tested people in the lab every semester, from years now, and I've had students that test positive for this, and they're like, oh my God, how come I don't have pneumonia? Mm-hmm. Well, one, it's not the only thing it causes, and two, you have a healthy immune system that can keep bacteria in check. Allow it to cause disease. So these are all virulence factors, so things that allow it to cause disease. One, it does have a capsule on it. So we've already talked about capsules this semester. Again, it allows that bacteria to hide from our immune system because it can't quite recognize it. It's also, just harder for those white blood cells to ingest it. It's kind of slippery. It has a protein called adhesin, and its name, adhesin, means it helps it stick like an adhesive. So these bacteria, again, before any bacteria can really start to grow and cause a disease somewhere, they first have to stick and find a home. And so the fact that they have a protein that helps them stick specifically in our respiratory tract, well, that's where the bacteria are going to grow and start to cause issues. We make an enzyme called secretory IgA protease, it's an enzyme enzynase, that breaks down your antibody A. So if you remember, we are immune system. We have 5 different classifications of antibodies or immunoglobulins. We have the A, I was going to say they were alpha, delta, gamma, mu, but IgA is one of our antibodies. So as your immune system tries to attack it, one whole group of antibodies, IgA, it breaks it right down. So you have a whole group of antibodies that can do absolutely nothing for this bacteria. And it has an enzyme that does an endogenase called pneumolysin that destroys or breaks apart epithelial cells. Our initial respiratory tract is lined with epithelial cells. And because it can break apart epithelial cells, this bacteria can gain entry into places that it normally shouldn't be able to. Again, helps it cause disease. Good things for it, all bad things for us. And so some of the diseases that it can cause is, yes, its name is pneumonia, and yes, it can cause pneumonia. Those that are at the highest risk, elderly, immunocompromised, and infants. Again, healthy immune system people, very rare that they would ever get pneumonia from this particular bacteria, and some people are even normal carriers of it. Although its name is pneumonia, that doesn't mean it's the only place that can affect is the lungs. If it gets into your sinuses, you have a sinus infection. If it gets into your ear, because that's linked to your respiratory tract, like the whole way, it can cause ear infections. It can get into the bloodstream and cause bacteremia. It can affect the heart and cause inflammation of the heart. It can get into your cerebrospinal fluid. It can cause meningitis. Lots of things, depending on where it goes. Now, my little note on meningitis, it's like my little side note with pneumonia. Pneumonia is it's a condition, not a disease, because there are so many things that can cause it. Bacteria can cause pneumonia. Viruses can cause pneumonia. Fungus can cause pneumonia, because we're going to talk about all these different things that can cause pneumonia. Pneumonia, again, just means there's fluid in the lungs. There's difficult breathing. Generic term. Meningitis is the same thing. The word meningitis means inflammation of the meninges, but there are lots of things that can cause that. There are bacteria that can cause that, viruses that can cause it, fungus that can cause it, parasites that can cause it. So a lot of times when we talk about meningitis, we usually put another word that goes along with it to help kind of narrow it down. So if I say pneumococcal meningitis, the fact that it has this coccal part of the word in it, right away it's going to narrow down. It's a bacteria that's cocci shaped. So we know, okay, pneumococcal, but we know it's not a virus. We know it's not a fungus. We know it's a bacteria. We know it's a cocci shaped bacteria, which is pretty much going to narrow down to a gram positive. It's very little gram negatives cause it. And the fact it's got the pneumo in it also leads it back to the pneumoniae bacteria. So my little side note is lots of things cause meningitis. Always pay attention to if there's another word that goes along with it that helps narrow down what actually is causing the meningitis. How we diagnose this bacteria? Well, we can gram stain it, usually a sputum sample, because it's coming from that respiratory tract and the pharynx. We can grow it out. We'll see some of the alpha hemolysis that it shows. We can also test it to see if it is sensitive or resistant to the antibiotic Optogen. And so we had a plate a couple weeks ago in lab that looked like this, that had Streptococcus pneumonia on it. And we had the Optogen that had the little P on the disc. And the bacteria could grow anywhere except where that Optogen antibiotic disc was sitting. You can also put this bacteria in with some bile. It's part of the digestive tract. It helps break things down. This bacteria will break down in bile. It's not normally evolutionary exposed to it, so it is affected by bile, unlike our next group of bacteria. So it's a couple ways to help narrow down this particular bacteria. It's treatable, penicillin. Otherwise, if that's not working, erythromycin is generally our next one to grab. But we do have a vaccine for Streptococcus pneumonia, and it's a very effective vaccine. I've been starting to see commercials for it now that we start getting into fall and everyone, people stay indoors and lots of stuff gets spread. It's really promoted to the elderly. Again, healthy immune systems. Doesn't mean you can't get vaccinated, but you're less likely to suffer any complications. So generally, elderly immunocompromised are really promoted, especially in the commercials, to get the vaccine. Now, our last group of still gram-positive cocci, but they're enterococcus, so they still would not have any bubbles with a catalase test. Based on the part of the word entero, anything entero means digestive tract, means intestine-driven. So an enterococcus is going to be our first clue that this is a bacteria that is found in the digestive tract. When we gram stain it, it's gram-positive, it's cocci. Also likes to hang out in short chains or pairs, which is going to be just like that strep pneumonia. So a gram stain can help narrow things down quite a bit. You know if it's gram-positive or not, you know what shape it is, and the arrangement can even help narrow it down to strep pneumonia or an enterococcus. And there's further tests to narrow it down. Although there are lots of species of enterococcus, there's only two that cause human disease, so they're the only two that we care about. It's Enterococcus faecalis and Enterococcus faecium. They are both normally found in the human colon. Again, they're Enterococcus. They're supposed to be found there. And they're part of the normal flora. Again, if they get somewhere else outside of that intestinal tract is when they can cause infections. Now, these two bacteria that are found in the digestive tract are also a couple of the top causes of healthcare-associated infections, also known as nosocomial infections. Again, they're getting away from the word nosocomial. I need to update this. And just call them healthcare-associated infections, because no one knows what the word nosocomial means. It just means these are two bacteria that commonly cause disease for people that go into a healthcare setting. So whether they go to the hospital and pick up this bacteria, whether they go to the hospital and have a procedure done, and then they all of a sudden have an infection with this bacteria, whether they go to even to a nursing home or any kind of healthcare setting, this is a bacteria that causes infections. And part of it is not because they're just hanging out in that facility. These are bacteria that are found in fecal matter, and it's sad to say, fecal matter is everywhere. People do not wash their hands. They touch everything. And you have, in healthcare settings, lots of unhealthy, immunocompromised, very susceptible patients that can pick up these bacteria that can then cause disease. Also, if people are going into healthcare settings, they might be having colonoscopies or other digestive tract procedures done, and that bacteria can get somewhere where it's not supposed to. So these don't cause a lot of infections just for the normal public. They're usually more linked to a healthcare setting that you went there and got some procedure done and then picked it up and caused issues. When we diagnose it, to distinguish it from one of those strep bacteria, that pneumonia, because they're going to look identical to each other under a gram stain, is we also mix it with some bile and look to see if it can grow or not. Enterococcus, because they are already found in the intestinal tract, they've evolved to live in amongst bile, because bile is present in the digestive tract. And so they're not sensitive to bile. They will grow on a plate that has bile added to it. Strep bacteria will not. Our problem with Enterococcus bacteria is they are one of the leading group of bacteria that is developing resistance to antibiotics at one of the fastest rates. And so, I'm trying to remember, there's a whole bunch of new acronyms now, not just the MRSA, that are all including the Enterococcus bacteria that are resistant. So people can go into the healthcare setting, have a procedure done, pick up this bacteria because it got somewhere it wasn't supposed to, and now we don't have anything to treat it. And so prevention is difficult because it is a normal flora, so it's everywhere. Our best bet is to minimize transition. Good hygiene, good medical practices, PPE, because it's out there. We have it. There's no way we can avoid it. We can't get rid of it. We can't give someone an antibiotic unless we kill every single bacteria in their body, which we can't. We can't get rid of the bacteria. Our best bet is just to minimize the transition so no one gets it. Now we're going to switch to a totally different shape. I don't want to go back. So away from gram-positive cocci and onto gram-positive rods or gram-positive bacilli. Now, of all the gram-positive bacteria that are bacilli or rod-shaped, we group them into two groups, whether they can make endospores or not. And there are two groups of bacteria, these should look familiar, two groups of bacteria that can make endospores, anything in the Bacillus genus and anything in the Clostridium genus. And so we're going to focus on those bacteria and look at just the top bacteria that can cause disease. Again, I'm only focusing on the top bacteria that can cause disease. Your books have more bacteria that can cause disease, and there are still more that the books don't even cover, but top bacteria that you may have heard of. Starting with the Bacillus genus. So when we gram stain it, again, they don't always gram stain the greatest because they can form endospores, but they are gram-positive bacteria if we did a full-on gram stain with them. To know if they can make an endospore or not, we would want to do an endospore stain. But Bacillus anthracis is really the only bacillus that causes human disease. There are lots of bacillus bacteria out there. They look pretty cool on plates because they usually have really big colonies. They're normal flora. Bacillus anthracis is the bacteria that can cause disease. It's not super common. And what allows it to cause disease is the fact it makes toxins called anthrax toxins. Now, most people pick up this bacteria from handling infected animals. There are lots of animals, farm animals, generally not pets, but farm animals that are carriers of this bacteria. Doesn't mean that they are suffering from it, but if you are in contact with them, so farm workers, you are at a higher risk. There are 3 ways to pick up the bacteria, and based on how you pick it up depends on what anthrax disease you get. You can either eat the spores, they get in through some type of open wound, or you inhale them. And so our 3 types of anthrax diseases are just based on which way you picked it up. So gastrointestinal anthrax means you ate some of the spores. It's extremely rare. It's going to cause lots of food poisoning symptoms, but it is deadly. Well, I shouldn't say it's always deadly. I think if it's left untreated, it kills about 50% of those that ingested spores. And if diagnosed and treated, it kills about 40% of those that did it. So like treating it's not going to increase your chances all that well. The more common way that it's picked up is working with animals that are carriers of it and it gets into an open wound. Once it gets into an open wound, those toxins do kill tissue and you end up with these black eschars. This is dead tissue that's now replaced with just like a black, dead material. It is generally not deadly. It's going to be painful. The more common way. Again, farm animals can be carriers of the bacteria. You work with farm animals, it gets into open wounds, any kind of cuts, whatever. You end up with these black eschars. Another way it can be picked up is you inhale the spores. Also extremely rare, also extremely deadly, because that bacteria is going to get in there and it's going to cause death of tissue in your lungs, which is not good. So it's also very, very deadly. This is generally the one when people think of anthrax, they think of bioterrorism. Oh, a suspicious envelope got mailed somewhere. And yes, you could breathe in spores, and yes, it can be deadly. However, the bacteria, if you know you're exposed, you can like pre-treat for it and start taking antibiotics just in case. And if you can catch it ahead of time, it's very treatable. But this is extremely rare that this would actually happen normally out in nature, that you would breathe it in. To diagnose someone with this bacteria, they can grow the bacteria on the plate. They can grow it out. Again, the colonies are usually quite large on a blood agar plate. They can gram stain it and see the fact that, it's these big, huge gram-positive bacilli. They could also do endospore stain. But there are lots of antibiotics that can be used to treat it. The biggest thing is getting treated before you start to have symptoms, before it starts to spread. Because people are generally not exposed to the bacteria, the best way for prevention is that we treat animals, so that farm animals are treated for anthrax. If the farm animals are treated for it, the farm workers then don't have that risk. There is a vaccine for anthrax for people that work for the CDC that will be working with anthrax, or maybe they're the people that are working with animals that can't be treated with any type of vaccine or treatment. So we do have a vaccine. It's not the most effective, which is why we're not really exposed to it. It generally doesn't cause us issues. It's not the most effective. It usually requires lots of boosters. But for those that are constantly exposed to this bacteria, we can vaccinate them as a preventative as well. Group of bacteria that can make endospores, so they're gram-positive, they're bacilli, they can make endospores, is the Clostridium. And there are 4 Clostridium bacteria that we're going to talk about that are very common and cause very horrible diseases. And I'm guessing you probably heard of at least 3 of them. Maybe not the first one. So when we gram stain them, we can sometimes see that they can make an endospore. They usually don't hang out in long chains. The little endospore itself, we kind of see it at the little end. It kind of looks like a little club. Again, it's better to do an endospore stain. But anything in the Clostridium genus, they are all anaerobic, which means they have to get into an anaerobic environment to grow. So they are not going to grow on the surface of our skin. They would have to get in and under the skin into an anaerobic environment. Anything in the Clostridium genus, they are found like everywhere. Some of these bacteria are found in soil. Some of them are found in water. Some of them are found in the digestive tract normally of animals and humans. And so these bacteria are everywhere. It's like you can't avoid them. Now, these bacteria also have a couple different types of enzymes that help them cause disease. One of them is called lipase, one's called protease. The end in ase. Lipases break down lipids. Proteases break down proteins. So these bacteria break down our structures. They break down cells. They break down lipids. They break down proteins. It helps them get where they need to go, but it also causes catastrophic damage to our own tissue, which is why they cause such horrible diseases. So the very first Clostridium that we're going to go through, which is the one you may not have heard of. I've had students who are like, oh yes, I know this bacteria well. I just didn't know the name of it. It's called Clostridium perfringens, and it has toxins in it that causes irreversible damage to the body or irreversible damage to the tissue. This is a bacteria that has to get in the body, and it has to get into an anaerobic environment for it to cause disease. And its top 2 diseases that it can cause. One, it is a top cause of food poisoning. So someone didn't wash their hands very well and touched food, and then they didn't refrigerate it, and bacteria grew all over your food. It can cause food poisoning. That in itself is not that bad. It will go away on its own. The worst part, though, is when it gets into tissues, skin tissue. So if there's an open wound, and this bacteria gets into that open wound, into an anaerobic environment, those enzymes will start to break down the tissue, and it will also release a very noxious, very smelly gas, like really stinky, and cause complete irreversible damage to the tissue. It's called necrosis, but that smell of it, because it releases that horrible gas, is called gas gangrene. So I've had students in the past that have worked in nursing homes. They've worked with diabetic patients where they literally are losing tissues. They don't have good immune systems, and all it takes, and it usually in the toes, especially with diabetics, is this bacteria gets into any kind of open wound, and it causes irreversible damage to tissue, and it is extremely stinky, like it's really smelly for this particular bacteria. Now, to diagnose someone with this particular bacteria, they do bacterial counts. If they think it's a food poisoning, they would look for the number of bacteria that's in the fecal matter to say that that bacteria is actually causing food poisoning. They can also grow it on a blood agar plate, and it has what's known as a double zone of hemolysis. You can kind of see it. So there's the big, huge colony. It has this very light colored hemolysis, but around the outer edge, it has almost kind of a pinkish or reddish color that's lighter than the rest of just a regular blood agar. That's a very unique distinguishing characteristic on a blood agar plate to know it's this bacteria. For treatment, if you suffered food poisoning from it, the only treatment is to let the food poisoning take place. There's a reason why if you have food poisoning, your top symptoms are vomiting and diarrhea. It is your body's way of the fastest way of getting rid of the bacteria. It's by vomiting it up or through the diarrhea. And the bacteria will leave on its own, and there's generally no long-lasting effects. Gas gangrene, if it causes that irreversible damage to the tissue, it is tissue removal. Whether it's full toes or just that actual surgical removal of the tissue, but you have to remove the tissue. So how to prevent it? Make sure foods are refrigerated. Make sure food's not sitting out and bacteria that do get in there get into high enough counts that would cause disease. And making sure wounds are cleaned properly, especially for those that are immunocompromised. Diabetics, they have very little blood flow going to those fingertips and toes, which makes that, even if there is an infection, they can't get the white blood cells to those areas. So they're very prone to infections in those areas. So making sure wounds are cleaned so that there's absolutely no bacteria in them that they have to fight off. That might be the one where maybe you haven't heard of it. Our next three Clostridium, you probably have. So the one is Clostridium difficile, also known as our C. diff. Now, again, there are so many Clostridium. We're just talking about the top 4 Clostridiums that cause human disease. And as I mentioned in lab, because there's so many Clostridium, they are now slowly starting to reorganize them. And because this one is found in the digestive tract, they really wanted to name it Peptoclostridium. But again, we didn't want to start calling things P. diff because everything's listed as C. diff. So now it's just called Clostridioids. So I listed as Clostridium, but don't be surprised if you start to slowly see, as people are making new labels of stuff, see it listed as Clostridioids. I don't know. Maybe it'll still stay Clostridium. It's just it's not really Clostridium, but it's Clostridioids. Again, there's just so many bacteria in this group. They were starting to slightly name them different. So C. diff is a bacteria found in the digestive tract. And for some people, it's part of the normal flora, which means you guys might have C. diff in your intestinal tract right now, and it's not causing you any issues. But it can become an opportunistic pathogen. And opportunistic meaning it doesn't normally cause disease, but it can. And it usually causes disease when you take a broad spectrum antibiotic. And it's a unique thing. Most people don't get a bacterial infection when they start taking antibiotics. It's normally the reverse. You take antibiotics to get rid of a bacterial infection. But a broad spectrum antibiotic that's taken for something like a sinus infection or an ear infection is going to kill some of your other normal flora, the other normal flora that are keeping this guy in check. And so when you kill the normal flora, this one's harder to kill. It is an endospore. This one starts to have itself a little party because there's nothing keeping it in check. It's like you killed the parents and the kids have a party. So this can cause anything from minor infections to explosive diarrhea. Your book actually lists it, and so I liked it. And it's because it has toxins that can cause damage to the digestive tract. And because it can cause infections and damage to the digestive tract, it can lead to something known as pseudomembranous colitis. Again, anything itis means inflammation. So this is inflammation of the colon. So the toxins that this bacteria can make will cause enough damage to the colon that the colon starts to slough off on the inside. It almost forms ulcers in the colon wall. They're going to be very painful, and your intestinal tract is just trying to get rid of the bacteria in the only way it knows how, and it's going to be through the diarrhea, and now we have damage to the colon as well. That's going to be very, very painful. Now, it's considered a nosocomial pathogen or a healthcare-associated infection, not generally because people pick it up when they go to a healthcare setting, because most people already had the bacteria to begin with. It's because you went to the healthcare setting and you were given an antibiotic. And that antibiotic killed your whatever it is that you were going in to get treated, and now you have this afterwards. So this is one of those that are a lot of times called secondary infections, is you killed the first infection and then this one showed up. Now, diagnosing it, we can isolate the bacteria from fecal matter, but generally if someone has a C. diff infection, they have diarrhea, which is going to be even harder to find the bacteria in there. Instead, we can do immunoassays. We can look specifically for the toxin. We can do antibody-antigen reactions. We can also do PCR. This is looking specifically for the DNA of this bacteria. It's usually faster. The treatment, it's like the reverse of like any other bacterial infection. The treatment is to stop antibacterial use. So if you're on a broad spectrum antibiotic to treat your sinus infection, your ear infection, or whatever other infection, we need to stop that. We need the normal flora back, like in as fast as possible. So we want to stop killing bacteria because we want them to come back. On rare cases, a really strong antibiotic known as metronidazole is given. Those are usually for very extreme cases, but that can sometimes cause more side effects than benefits. One of the newest type of treatments that's out there is doing a fecal transplant. Again, normal flora keeps this bacteria in check, and we know that. There are good bacteria that just don't get any credit. There are good bacteria. Well, we can take those good bacteria that were from someone else's feces and then put them in the digestive tract of someone else, and those good bacteria will keep this bacteria in check. This just started probably within the last 5 years, becoming pretty common. Even when I started here, this was just like in, experimental. Was it working? And now it's actually becoming quite common because it is super effective. For people that are suffering from a very bad C. diff infection, they will do fecal transplants, and it's almost within 24 hours, it's night and day. A patient that was on their deathbed all of a sudden is just totally fine and healthy. It's because those normal flora bacteria kept this bacteria in check. The biggest issue they were coming up with is how to bill insurance. Because we're like, is it like an organ transplant? Is it like an antibiotic? I'm not sure even still what they coded it as. Best prevention, good hygiene, so that if you don't already have the bacteria in the body, we don't get it into a patient. And then also probiotics. For people that are taking any type of broad spectrum antibiotics, a good doctor will tell you, you're on a broad spectrum antibiotics for your ear infection, nose infection, whatever it is. You should be eating yogurt and taking probiotics just to try to keep those good bacteria coming back and healthy so that you don't suffer from one of these secondary infections. Our third Clostridium is Clostridium botulinum. And this bacteria causes the disease known as botulism, which I talk more about on my next slide. But I'm going to do like my little refresher of how does a nerve or a neuron cause a skeletal muscle to contract. So I think in A&P, this is like an entire lecture. I spend almost an entire lecture on it when I used to teach A&P. When you have a neuron and you have your axon terminals and you have information coming from your brain and it is traveling on those axon terminals, at the end of the axon terminal, you have these little vesicles with acetylcholine. They cross the synaptic cleft. They bind on your skeletal muscle. There's your skeletal muscle. And they get to one of those sodium-potassium channels, and they eventually cause calcium to move, and your muscle will contract. It was like a 4-minute lecture in A&P. So our skeletal muscles contract because our nervous system tells it to contract, because this acetylcholine can cross the synaptic cleft and carry that message. Muscles need to contract. What Clostridium botulinum does, it has toxins, those botulism toxins, that cover the end of our axon terminals, which means we can be telling our nervous system, telling our skeletal muscles to contract, but if this acetylcholine can't leave the axon terminal, cross the synaptic cleft, and get to our skeletal muscle like it's supposed to, because these toxins are blocking it, your skeletal muscle can no longer contract. That's an issue because we have a diaphragm that is made up of skeletal muscle, and it's your diaphragm that allows our lungs to change volumes and allows us to breathe. If your diaphragm can't contract, what will happen to you? You will suffocate and die. It's a very deadly toxin. Now, extremely deadly because it causes muscles to no longer contract. Skeletal muscles, that means we can't breathe. Now, there are 3 ways we can pick it up. And so then there are 3 different kind of outcomes or 3 different types of diseases. One is called foodborne botulism. It just means you ingested the toxins. Doesn't always mean you ingested the bacteria itself and you have an infection of bacteria, but you ingested the toxins. And if you ingest the toxins are now in your body and they're going to do exactly what they want to do. And they're Going to stop all of your skeletal muscles from contracting. Botulisms, because this bacteria, it's Clostridium, it's anaerobic. It needs to get into an anaerobic environment for it to grow. And it is not uncommon that this bacteria gets into food products. And when they're in an anaerobic state in the food products, it will grow. The bacteria will grow. It will make all their toxins. And then you ate the food and you ate the toxins. One key thing to know that your food is infected with some type of bacteria is if you ever see a bulging can, meaning somewhere in the canning process, a bacteria, whether it's this one or not, a bacteria got in. And I'm going to say any bacteria could potentially get in. Most bacteria are killed in the canning process. This one, because it can make endospores, is harder to kill. It may not have gotten killed in a canning process. And because it's growing, cans will start to bulge out. Don't ever buy a bulging can. Don't ever even open it. Don't ever try it. It's not good. Some of the initial symptoms, if you did ingest it, usually within a couple days, again, it's starting to damage skeletal muscles. You usually start to have dilated pupils within a couple days. You're usually going to have constipation. You're usually going to have lots of abdominal pain. I'm trying to think of some of the other. I feel like there was another one. I'm not going to look it up. But it's normally enough where you're like, something's going on, you go to the hospital. Problem is, a lot of times by the time you are diagnosed with an infection, it could be too late, that the damage is already done to certain skeletal muscles. Infants are actually the number one group of individuals that suffer from botulism poisoning, and it's because they ingest the bacteria, not just the toxins. We can actually ingest some of these bacteria, and our immune systems can get rid of them and keep them in check. Infants, if they ingest this bacteria at all, they do not have a strong enough immune system to kill the bacteria, which means the bacteria will take hold, they will make those toxins, and it becomes deadly for them. The number one place that infants have actually picked up this bacteria is from eating honey. This is why if you ever look at the back of a honey label, and you will now, it will always have a label on there. Do not feed honey to infants under the age of 2, I believe it says. I can't remember if it's 1 or 2. And it's because honey can carry this bacteria. Honey in itself is antibacterial, which means they don't have to go through a whole bunch of treatments. They don't have to can it. They don't have to get it up to a certain temperature. Bacteria don't grow in honey, but they can stay dormant in honey if it's a Clostridium that can make endospores. And if infants eat honey and they eat the bacteria, they can suffer from an infection. We would not. Our immune systems would just kill the bacteria right away. That's why you see the label on there for honey. This bacteria can also get into wounds. Again, if you have any type of open wound, it can get in there. It's going to cause all the same effects as whether you ate it or not. It usually just takes a little bit longer. But if there's any kind of open wound and this bacteria is on any surface, it can get in there. Diagnosing it, any of the symptoms, usually dilated pupils, constipation, abdominal pain. I want to look it up. I know there was at least one more symptom I wanted to mention. Blurred vision, I guess that goes with the dilated pupils. So treatment, if they suspect a patient has ingested this particular bacteria or the toxin, the treatment is giving antibodies against the toxin. Again, your immune system, it could recognize the bacteria, it could recognize the toxin, it could mount a whole immune system, that whole flow chart that I drew. It could do all that, but it could take a couple weeks, and you might not have survived those couple weeks. So they will try to give you the antibodies that will bind to the toxin and try to neutralize the toxin. And then hopefully your immune system can kick in and do the rest. They will also give you antibiotics to try to kill any bacteria that's present. So the bacteria is not making any more of the toxin. The big thing is to neutralize the toxin. The toxin is what kills, because it's the part that stops that whole muscle from contracting. Prevention, proper food canning, and no honey to infants. Now, I know you guys are a small group, but do any of you can anything? I was gonna say, I'm always like, whatever, in college I didn't can. I'm like, I do now. So this is the, how you can is literally the canning process is specifically fitted just to prevent this bacteria. So when it says to boil it for 15 minutes or to pressure cook it for 20 minutes, it is specifically tailored to prevent this bacteria. Because again, it's an endospore bacteria, it's harder to kill. So if you can kill this bacteria, you're pretty much good that it's sterile, you've killed everything that's possible. So proper canning, there are two ways to can things, boil canning and pressure canning. Pressure canning, just like our autoclaves, you can get up to a higher temperature under pressure for a shorter period of time, and it's going to kill more. But it's putsy and it's a pain, and people would rather not do it. So to kill this particular bacteria, Clostridium can be killed using a boiling method if it's also an acidic food. So if you have acid and boiling, 100 degrees Celsius, acid and boiling will kill this bacteria. And so foods like fruits are naturally acidic. Tomatoes are naturally acidic. You can boil can them because they have acid in them, and so it will kill this bacteria. Other foods like meats that are not acidic and various vegetables that are not acidic, you have to pressure can them, is because they don't have that acid. Although in a lot of canning procedures, most of them, it's one of the very first steps is add lemon juice. Any guess why? It has the acid. Although the fruits may even have acid in it, let's add lemon juice to give it more acid so that you can still boil can it, and it'll have that low enough pH plus the boiling to kill this bacteria. So it's all tailored just to kill this bacteria. Now, the botulism toxin is the thing that causes skeletal muscles to not contract. But this particular toxin is used elsewhere. You've probably heard of before. Botox? Heard of it? Uh-huh. What's Botox for? What is it used for? Yes, for what purpose though? Wrinkles. I have a ton of wrinkles, especially on my forehead. It gets wrinkly because you are constantly contracting muscles. Every time I contract my muscles, my wrinkles show up. But if I put the toxin underneath my skin right there, that toxin is going to prevent my skeletal muscle in my forehead from contracting, which means the wrinkles will go away for a short period of time. That's what Botox is we are injecting this particular toxin right underneath the skin to make it so that your skeletal muscles can't contract in your face, and that magically your wrinkles will go away. It's awesome. However, that also means you have like no facial expression. If they put a lot of it in there, because you can't contract your muscles, you can't smile, and you can't, you just can't do facial expressions because your entire face is like frozen, but it will have less wrinkles. But that's what Botox is. And so you hear a lot of commercials for Botox. Listen to them next time as they start to say the little side effects that could happen. And if you start to have trouble breathing, it's like, that's because in theory, when you inject it right underneath the skin, it should stay right underneath the skin. Should. However, it could get into the bloodstream and it could travel elsewhere in the body. And if it travels to the respiratory tract and gets to that diaphragm, can't contract, you're going to have difficulty breathing. So that's why I'm like, listen to the side effects. And it's like, it could cause this or it could cause this. And then you should see your medical professional. You are injecting a toxin that, yes, is very, very deadly. But if it's put in just the right spot by a reputable person, it should stay in place. Should. Doesn't mean it will, which is why they have the side effects listed. It's a little interesting of why we use, this. Why we use bacteria for cosmetic purposes. Now, a side effect is they specifically did target Botox just for wrinkles because it stops those skeletal muscles from contracting, but they had a secondary benefit of it, and it was for migraines. And it was like, well, all of a sudden, a lot of your skeletal muscles contracting would trigger migraines. Well, if we stopped the muscles from contracting in the head and the face, it actually decreased the number of migraines that people had. And so it all of a sudden started to become a migraine treatment as well. All right, away from botulism, get into our last Clostridium, which is Clostridium tetani, which causes tetanus. Again, it's the toxin that causes the issues, not the bacteria itself. And just like botulism, it targets skeletal muscles. It targets the nerves controlling the skeletal muscles. But it works in opposite of botulism. Botulism prevents a skeletal muscle from contracting. The tetanus toxin prevents the muscle from relaxing. So we have various acetylcholine. We have various things that go from our nerves to skeletal muscle that tells our muscle to contract, but we also have chemicals that tells our muscle to relax. Like, okay, I'm gonna like contract my muscle, but then I wanna relax it. And we've got chemicals that tell us to do both. Well, this toxin prevents the release of that inhibitory neurotransmitter, which means your muscle will contract and it will stay contracted. It can't ever relax. That toxin prevents the relaxing. Now, again, this means it can affect that diaphragm, which is a skeletal muscle that's needed for survival. If your diaphragm stays in a permanently contracted state, what will happen to you? You will suffocate and die. Again, very deadly. They just work in opposite ways. And so one of the very first diagnoses is that this particular bacteria starts to kind of affect skeletal muscle. But one of the very first skeletal muscles that seems to be affected are the muscles in the face, particularly the muscles in the cheeks, and it causes something known as lockjaw. Is your skeletal muscles, when they contract, your mouth closes because it makes that a smaller muscle. When it opens, it's because the muscles are relaxing to allow it to open. Well, if they can't open, if they can't relax, your mouth stays in a permanently contracted state. And so that's usually one of the very first symptoms. I mean, usually there might be twitching. I shouldn't say the very first symptoms. All of a sudden you might have involuntary twitching. Your skeletal muscles are starting to contract. They're starting to get affected. But ultimately, it can cause all of your muscles to stay contracted in a permanently contracted state. Not just your mouth, your diaphragm, all of your muscles. They say it can cause your muscles to contract so hard it can break internal bones because those muscles are contracting with all the force that's not controlled by your brain anymore. So treatment, the very first treatment is to get the antibodies against it. We have to stop the toxins. You know, it's the toxins that are causing the damage. We need to stop the toxins ASAP. And so it's to get the antibodies against the toxin. Then it's to give some antimicrobial drugs to kill any bacteria so they don't make any more toxins, but it's all about the toxins. And then they can also give you an immunization so that, again, it also increases your immune system at trying to build up your own antibodies against the toxin. Now, I found out it was a couple years ago. This is not a super common infection around here. Again, most people are vaccinated for it, but there is one big group of individuals that live in our area, in the Wisconsin, Minnesota, Iowa area, that generally are not kept up on routine vaccines, and it's the Amish. And it was a couple years ago that Gunderson had an Amish person that came into Gunderson Lutheran that was starting to have the twitching, starting to have some of these involuntary muscle contractions, and they were able to diagnose him. The unfortunate part is that, you know, by the time you diagnose, and generally by the time you have symptoms, it's almost too late, that they had to put him into a medically induced coma. And I also found out too, Gunderson does not carry antibodies like on stock against this. They had to get him shipped in, which took like a week, which that also shocked me, that it took that long. They were eventually able to treat him, but he was in the hospital for like 2 months. He survived, but most people don't. Generally, by the time you have symptoms, it's usually too late for treatments. So the best is to prevent it, making sure you're updated on vaccines. Now, when do most people think they should get vaccinated? When, I should say, I'm like, how do most people think they're going to pick up tetanus? It's like rusty nails, rust anything. It's not to say that rust won't do it. This particular bacteria is normally found in soil. And so any open wound that now has dirt in it, that's your entry source. It's an anaerobic bacteria. So it needs to get into an open wound, like into the anaerobic area, and it just has to be something dirty that gets in there. Now, if something's rusty, that usually means it's exposed to the environment, it's exposed to dirt, dust, anything that lands on it. But really, any cut, if you are working in the garden and you've got an open wound, you are at high risk of this more than just rust. It's like any open wound working with soil or dirt puts you at a higher risk. Our last group are the gram-positive bacilli that don't make endospores, and there's 4 of them. So the first one is Listeria, and there's only one Listeria that causes human disease. It's Listeria monocytogenes. So it is a bacilli. It's a coxobacilli. So it's like a short little rod. And the number one way we pick it up, contaminated food and water, or food and drink. I shouldn't just say water. Listeria's claim to fame on how it survives and causes disease, it is an intracellular parasite. It will actually cause our cells to ingest the bacteria. It just can't get broken down once it's in there. So it hangs out, growing like crazy, hiding out in our own cells. This is showing an actual picture of the bacteria getting uptaken into our cells. Its virulence factor that helps it cause disease is the fact it is living inside of our cells. What it can cause, it causes meningitis, a very deadly meningitis. That usually causes death in fetuses and immunocompromised. Healthy individuals maybe suffer some food poisoning symptoms, but that's usually about it. And it causes it in a fetus, which means this is one of the few bacteria that can cross the placental barrier. So if mom eats something that was contaminated with this bacteria, it can kill the fetus. So the biggest people that have to worry about it, elderly, immunocompromised, and pregnant women. Now diagnosing it, it is a flagellated bacteria. So in a broth, it will actually tumble around in like a big old circle. So it's interesting to look at to help it diagnose. Otherwise, we can culture it out. We can gram stain it. It'll help us easily see that it's a coxobacillus, that's a gram-positive. There aren't that many that cause disease. There are lots of antibiotics that can treat it. But because it is found in a lot of foods, it is a top cause of food recalls. And I want to say there was just one recently, too. I don't, and I'm like, and it's when sometimes I see them and I'm like, I'm going to share this. I'm like, wait, we haven't even talked about the bacteria yet. I'm going to look it up. It wasn't that long ago that Bluebell had a big recall for Listeria. I'm trying to think of other things. But don't be surprised. You'll hear recalls now for Listeria. Again, the biggest foods that are at the highest risk that if you're pregnant you want to avoid altogether, Brussels sprouts, certain deli meats, raw milk, soft cheeses like bries and mozzarellas, and smoked seafood are the top normal locations where we can find Listeria. Second genus that's also gram-positive bacilli, that's not endospores, are the mycoplasms. They're the smallest free-living microbes, which means they can just like be found out in the environment. They don't have to grow in us, but they can. An interesting part is that these bacteria cannot make peptidoglycan. They can't make a cell wall. They are still genetically gram-positive, you know, saying they have a thick cell wall. Genetically, they're gram-positive, but they can't make the cell wall. And so they are pleomorphic, which means they can vary in their shape. They don't have a nice thick cell wall that gives them a shape. So some might be a little more roundus. Some of them are more rod or kind of a coxobacillus shape. It usually requires some different growth factors. They're kind of picky on how to grow them. In us, we usually find them in the respiratory tract, the urinary tract, and they are not top causes, but they can cause pneumonia. If it gets into the respiratory tract, they can cause urinary tract infections. The most common mycoplasma is Mycoplasma pneumoniae. That when it gets into the respiratory tract, it's known as atypical pneumonia, or just more commonly known as walking pneumonia. you guys known anyone that's had it, or if you had walking pneumonia? I was gonna say, I'm like, it's, again, you'll start to hear people that are like, oh, I have walking pneumonia. It's a pneumonia, but it's called walking pneumonia because it's not like a normal pneumonia where you'd be in the hospital because you're struggling from breathing. It is causing issues in your respiratory tract, but they're very mild symptoms. This bacteria is super slow to grow. So the symptoms, are just generally not super strong symptoms right away. There's usually no white blood cell response, so your immune system is not like, oh, we really need to do something. It's just like, whatever, it'll go away, and it does. So it causes a very mild disease. So it's called atypical pneumonia because it doesn't behave like a normal bacterial infection. It's usually spread by nasal secretions. Usually the top group of individuals that suffers or gets the most walking pneumonia are kids between ages of 5 and 15. They'll have a little bit weaker immune system. They seem to wipe their noses on stuff. We can diagnose it with an ELISA. Best prevention, since it's spread by nasal secretions, washing hands, good hygiene. Easiest way to prevent it. It is treatable. We have antibiotics that can treat it to make it go away, but sometimes they just, it'll go away on its own. It doesn't cause any serious complication. It's more of an annoyance than anything else. Our third of our non-endospore gram-negative or gram-positive bacilli are the Corynebacterium. They're also pleomorphic. Some of you actually identified your bacteria in your throat to Corynebacterium, that you had some of the pleomorphic gram-positive bacilli. You just didn't have this particular disease-causing one. So there are lots of normal skin and oral flora. That's why you may have diagnosed the bacteria you picked from your throat to a Corynebacterium. Most don't cause disease in this group. The one that does is Corynebacterium diphtheriae. Causes the disease diphtheria. And it's the toxins that cause damage. But one of the things that it can cause, the toxins can start to cause like a congealed ball at the back of the throat, known as a pseudomembrane. And it's that pseudomembrane that can actually cause individuals to actually suffocate, because it can literally block up the whole back of the throat and airway. For diagnosing it, you can visually see the pseudomembrane. Otherwise, we do have plates that are very specialized to just grow this bacteria. Treatment is because the toxin is what's causing the damage. We do have antitoxins. We do have antibodies against the toxin. Otherwise, we also give antibiotics, and if that pseudomembrane is large enough, it may require surgical removal. Now, the number of cases of diphtheria has gone way down. There used to be thousands, hundreds of thousands of cases of diphtheria every year. Now we're lucky if we have a couple every year. And it's because we have a very effective vaccine. It is the D in the DTaP vaccine. It's the diphtheria toxoid vaccine that our body can recognize that toxin and neutralize it right away. I'm going to say our last group of bacteria, or I guess the last group. I want to see if I can get through tuberculosis in a couple minutes. Again, still gram-positive, but the last group, Mycobacterium. Mycobacterium bacteria, we look for this, or there's different types of tests we can do this in lab. They are gram-positive, but these are the whole genus of bacteria that have that mycolic acid in their cell wall. That's that lipid that doesn't gram stain well. So they are technically gram-positive, but they do not gram stain well. To fully diagnose, we would have to an acid-fast stain, and we're looking for that pinkish color versus the nice royal blue. Anything in this genus, the unique characteristics, they're super slow to grow. They do have protection from lysis, meaning if they were eaten by a phagocyte, one of our white blood cells, they don't get broken down. It's great for them. And so they will grow inside of our cells. So they are intracellular. They hide out inside of our cells. And they're starting to become very resistant to antibiotics. Great. Now, there are, I think I go through 3 Mycobacterium. The top one that you may have heard of before that causes the biggest issues of the Mycobacterium is Mycobacterium tuberculosis, or more commonly known as TB. It's a pulmonary disease, and based on its progression, we say you either have primary, secondary, or disseminated tuberculosis. Primary tuberculosis means it gets into the lungs and it stays. Wherever it got in is where it stays. So it's like a primary infection. So this bacteria, here's the little red bacteria, they're getting eaten by our white blood cells and then they live in them because they don't get broken down. And they will grow. And as more white blood cells get recruited, they'll just sit there and get into more white blood cells. And they will start to congeal together and form a little mass called a tubercle. That's fine. That's still all primary tuberculosis. They're just growing and hiding out inside these big old tubercles, these big growths inside of our lungs. It's only when those tubercles start to rupture and it spreads to another area of the lungs, still lungs, it's considered secondary tuberculosis. So primary tuberculosis, it just grows and stays put and really isn't usually going to cause a ton of issues right away. It's only when it spreads we call it secondary. When it spreads from the lungs to other body areas, we call it disseminated, meaning it's not just a respiratory issue anymore. Now, when you have big growths in your lungs, where you should have lots of gas exchange, it's going to cause lots of issues. You're going to have difficulty breathing. It can cause enough damage to get into blood vessels. It can cause bleeding. It's why it's people that have TB could be coughing up blood, because again, they have lots of damage to that lung tissue. Diagnosing it, look for an acid-fast test. We can grow it on plates, we can do genetic tests, but we can also do different types of skin tests, or we can even do chest x-rays and look for those big growths. Now, do you guys all have to have your TB test yet already? As I say, some of you, if you are a CNA, before you go out, you have to have a TB test done. And what they do for a TB test, if you were ever wondering, they do actually put a little bit of the tuberculosis antigen. It's not the entire bacteria, but it's a little bit of the bacteria antigen underneath your skin. And what they're looking for is if you have antibodies to this bacteria, which means you've been exposed to it, you're going to have a reaction. It's an antibody-antigen reaction, which most people, I don't know if anyone that has had that reaction that's had their TB test done, but that would be a positive TB test. Then they would do further testing from there. But that's it. Treatment, it's treatable, but it's a hard bacteria to get rid of. It's usually a combination of antibiotics, such as these. These are really strong antibiotics that are targeting that mycolic acid, and you're usually on an antibiotic for a few months. It's not just like, you're 10 to 14 days and you're fine. Biggest prevention, we do have a vaccine for people that are at high risk. It's just not a common vaccine that's given out. Otherwise, face masks are going to prevent spreading that bacteria from coughing. Good hygiene, I think that was my last slide. I want to get to that. I will tape these last 2 slides that are left and put it on there because we're out of time. But I still need to collect your worksheet because I was going to do that at the beginning of the lecture and I forgot. Let me stop this recording.